Hello, we shall be discussing on the regulation of gene expression. Here we shall discuss on how the protein synthesis is regulated. For this we shall study a model called as the Lac Operon model. This model, that is Lac Operon, was first explained by two scientists named Jacob and Monod while studying on the lactose metabolism in the bacteria named E. coli. They explain that the lactose catabolisms is carried out by three enzymes named beta-galactosidase enzyme, Lac permease and transacetylase. The DNA segments for these three enzymes are located in the Lac Z, Lac Y and Lac A. So these three genes, they together form the structural genes. These structural genes is under the control of a regulatory gene. So this is the regulatory genes. So the structural genes together with the regulatory genes, it form the operand, and the operand model for this lactose metabolism, lactose catabolism, is called as the Lac Operon. Now Lac Operon consists of following genes. The first one is operator genes. This operator gene is indicated by O. This is O. This operator gene is lying just adjacent to these structural genes, and it controls the transcription of mRNA along these structural genes. Now this structural operator gene is under the control of the repressor molecules that will be produced by these regulatory genes. Now the second is the promoter genes. The promoter gene lies just adjacent to this operator, that is, this is the promoter, and this region is the region where the RNA polymerase enzyme will bind and will initiate transcriptions along the structural genes. This promoter regions is under the this promoter regions controls the rate of mRNA synthesis. Third is the regulatory gene. This is the regulatory gene indicated by Lac R. Regulatory gene, this region lies just outside the operon, and it produces a repressor that will bind to this operator genes and will suppress the transcription of the structural genes. So this is about the genes that are involved in the Lac Operon model. Now we shall be discussing on the functions of this Lac Operon. The function of this Lac Operon was explained by Beckwith in 1967, Epstein and Beckwith in 1968, and Martin in 1969. They explain in two ways. One is that in the absence of inducer, that is lactose, how does this Lac Operon functions in the absence of inducer, that is lactose? When lactose is absent, the repressor regulatory gene will produce a repressor proteins which will bind to this operator, and once this operator bind to this once this repressor protein bind to this operator, it will prevent the RNA polymerase to transcribe these structural genes, and transcription is being blocked because of the attachment of the repressor protein to this operator. Now in the absence of inducer, that is when lactose is present, in the second case, the repressor protein will produce the regulatory genes will produce a repressor protein, but due to the presence of this inducer, that is the lactose, this inducer or the lactose will bind to this repressor and it will inactivate the action of this repressor protein. Hence this operator remain free. Therefore the RNA polymerase will bind to this promoter region, and it will induce transcription along the entire length of this structural gene. So in the absence of inducer, the operator is blocked, therefore transcription is not proceeds. But in the presence of inducer, the repressor is being inactivated, and therefore because of the inactivation of this repressor, the mRNA bind to this promoter and transcription takes place. So this is how regulation of gene expression is being done.